Hi, my name is Boberski, and I studied the effect of diet composition on lifespan and colorectal cancer progression in the Drosophila midgut. So why did I study colorectal cancer? Well, colorectal cancer is the third most frequently diagnosed cancer worldwide, but the second leading cause of death. I'm particularly interested in early onset colorectal cancer, or colorectal cancer in patients under 50. This has risen steadily since the 1990s and now accounts for 10% of new colorectal cancer diagnoses. These trends in early onset colorectal cancer are currently unexplained. Only around 20% of early onset colorectal cancer cases are attributable to genetic hereditary cancer syndromes. That means that the remaining 4 out of 5 early onset colorectal cancer patients don't have a genetic smoking gun explanation for their cancer. However, there are some clues as to what has caused these trends. Early onset colorectal cancer rates have risen more in developed countries, meaning that they may be explained by environmental or dietary factors like the Western diet, which is high in fat and low in fiber. In these figures, I summarize the proposed impacts of dietary nutrients like fat and fiber on colorectal cancer progression. High fat diets have been suggested to cause dysbiosis, inflammation, amplify oncogenic signaling, and increase stem cell proliferation, all of which contribute to an environment that favors colorectal cancer progression. High fiber diets have been suggested to increase microbial diversity, reduce pathogenic bacterial load, increase the production of beneficial metabolites like short-chain fatty acids, and reduce inflammation, all of which contribute to an environment that reduces the risk of colorectal cancer progression. I studied colorectal cancer using a transgenic Drosophila model to evaluate the impact of specific nutrients on cancer and gut health. This model was gifted from the lab of Dr. Andrew Casali at the University of Lleida in Spain. It has, as you can see in the figure on the right, to generate tumor-bearing flies, I crossed a transgenic stock with the RAS oncogene activated with another transgenic stock with the APC1 and APC2 tumor suppressor genes inactivated. These genes reflect actual mutations in homologous cancer genes in humans, and in the parent fly stocks, they're maintained with a balancer chromosome so that these transgenes are non-lethal. To generate actual tumor-bearing flies, I crossed these RAS and APC stocks, and I performed mosaic analysis with a repressible cell marker to select tumor-bearing flies. This entailed a heat shock, and the flies that survived the heat shock were guaranteed to have the APC1, APC2, and RAS transgenes, indicating that they were tumor-bearing. I then placed these tumor-bearing flies on standard, high-fat, and high-fiber diets. For the high-fat diet, I supplemented the control diet, the Bloomington Standard cornmeal diet, which I use as my control medium, with 10% coconut oil. For the high-fiber diet, I supplemented this control medium with 10% psyllium husk, the active ingredient in Metamucil. I monitored these tumor-bearing flies over the course of their lifespan, and after 7 days and 21 days, I performed a midgut dissection and fluorescence microscopy, which I'll talk more about later. I anticipated that flies on high-fat diets would have a shorter lifespan and more tumors compared to their counterparts on high-fiber diets. Now, let's move on to my results. In my lifespan assay, I analyzed the number of flies alive over time on each dietary group. In the graph on the left, you can see the percentage of flies alive on the y-axis over time on the x-axis. In black, you see the flies on the standard diet. In red, the flies on the high-fat diet, and in blue, the percentage of flies alive over time on the high-fiber diet. The general trend here is that flies on the high-fat diet are dying sooner. That line is falling faster than their counterparts in the standard and high-fiber diets. And this was borne out when I did statistical analysis of this data. Using a Mann-Whitney t-test, I compared the high-fat and control groups and found a p-value of 0.047, which is less than the critical value of 0.05. This means that I can conclude with a 95% confidence that there's a statistically significant difference between these two groups. By contrast, when I was comparing the high-fiber and control groups, I found a p-value of 0.143, which is greater than the critical value, meaning that I can't conclude that there's any significant difference between these groups. I did this data analysis in R, a statistical programming language, which means the code is reproducible and it's not just like in an Excel spreadsheet on my computer or something like that. Now, my fluorescence microscopy assay, which is for me the most exciting part of my study, um, in my transgenic model, tumor clone cells are marked with green fluorescent protein, or GFP. That means when I dissect the midgut, which is the region of the gut in which these transgenes are expressed, I can see tumor clone cells glowing green under a fluorescence microscope. And I perform this assay on flies from each dietary group to analyze tumor burden. In the upper left-hand corner, you can see a parent fly, one of the RAS flies. And remember, this RAS transgene was maintained against a balancer chromosome, so this fly would not develop tumors. And that's exactly what I saw. There's no fluorescence in the tissue of the gut itself, only some autofluorescence in this tracheal structure. However, compare that to the standard diet, tumor-bearing sample from the standard diet in the upper right-hand corner. You see punctate dots of fluorescence, which represent tumor clone cells, and I imaged this sample at the Yukon Advanced Light Microscopy Facility. I counted approximately 20 tumors. In the high-fat sample, on the bottom right, there was a very similar presentation. I counted approximately 18 tumors, and that you can see similar, punctate dots of fluorescence, even though this gut sample was torn. However, there's a very different presentation in the high fiber sample in the lower right-hand corner. I only saw 4 tumors and a very different, none of those punctate dots of fluorescence, only some autofluorescence from the trachea. Now, what's really interesting is that these results are borne out in the work I've done this year with a larger sample size to try and further investigate the relationship between diet and cancer progression. These are midgut samples that I imaged this year at the Fairfield University's core facility fluorescence microscope. In the standard sample, which you can see on the left, you see the same pattern of punctate fluorescence, and you can see the entire midgut because I'm using a more advanced microscope. I analyzed this sample using Fiji Image J, and you can see the binary image I created using this program on the upper left-hand corner. And using this Fiji Image J, I counted approximately 90 tumors. And so there's a big, I'm counting more tumors because I'm using more exact quantitative methods, like Fiji Image J, to count these samples. In the high-fat sample, which you can see in the right, there are obviously lots more dots of fluorescence, lots more tumors, and I counted approximately 214 tumors using the same binary image that you see in the upper left-hand corner. So this is a difference that I'm observing this year with a larger sample size that I didn't really see last year because I had fewer samples and less advanced microscopy, is that there's actually more tumors in flies on high-fat diets, I believe, than flies on standard diets. This is my sample from a high-fiber diet from this year. There's a lot of autofluorescence in the lower left-hand corner because of the sample drying up. You can see the mounting medium, sort of that front retreating around the sample. This is a difficult assay to do because I have to dissect the fruit flies, under a dissecting microscope using forceps and a scalpel at my high school, mount them, and then bring them to Fairfield University, where they've often dried up before I can get good pictures of them using the microscope. However, even with this autofluorescence, you can still see a similar presentation. There are only 3 tumors in the tissue of the gut itself, I believe, and you don't see that same concentration of punctate fluorescent tumors that you see in these other samples. This preliminary data appears to support my hypothesis and merits additional study, and analysis with a larger sample size this year is still ongoing. Now, why is this important? Well, evidence for the impact of dietary fat or fiber may suggest causes for rising early onset colorectal cancer rates, pointing to specific environmental or dietary factors that influence the progression of cancer in those 4 out of 5 early onset colorectal cancer patients without a single genetic smoking gun cause for their cancer. Furthermore, this data contributes to the base of evidence for dietary interventions in colorectal cancer treatment. For example, if I saw a beneficial impact of high fiber diets on reducing the tumor burden, this might suggest that high fiber dietary interventions would be beneficial in colorectal cancer treatment. This year, I'm using immunostaining to investigate the mechanism by which diet influences cancer progression by measuring the activity of cancer signaling pathways like wingless or WNT. So how I'm doing this
2 minute
is
warning.
I'm taking the midgut samples that you saw earlier, and I'm incubating them in primary and secondary antibodies against armadillo, a transcription cofactor in the wingless signaling pathway. I can then observe these immunostain samples using a fluorescence microscope um, and see the localization of armadillo, this transcriptional cofactor, whether it's accumulating in the nucleus, suggesting that transcription of wingless target genes is being activated, and it's having these oncogenic effects on cell proliferation. Um, and so with this work, I hope to suggest a potential mechanism between uh, the intake of dietary nutrients and the development of colorectal cancer tumors in the midgut of these flies, and ultimately to help provide answers for early onset colorectal cancer patients. Thank you so much for your time, and I'm ready for your questions.